What ancestral forces allowed a rare recessive mutation to persist and concentrate within specific human populations for centuries? This question leads directly to Tay Sachs disease, a severe neurodegenerative disorder caused by reduced hexosaminidase A activity. Elevated carrier frequencies within Ashkenazi Jewish lineages reflect the combined effects of historical population bottlenecks, limited gene flow, and founder events. The condition originates from pathogenic variants in the HEXA gene, which disrupt the breakdown of GM2 gangliosides inside neuronal lysosomes. As GM2 accumulates, neuronal function declines in a predictable metabolic sequence. Explaining how this single enzymatic defect became geographically and ancestrally patterned requires examining genetic drift, settlement history, and long intervals of demographic isolation. Hexosaminidase A operates within lysosomes, where it assists in the stepwise breakdown of GM2 gangliosides. The enzyme requires both A and V subunits to maintain full catalytic activity. Mutations in the HEXA gene impair formation of the A subunit, reducing the enzyme's ability to process GM2. However, residual activity varies depending on the specific amino acid substitution or structural disruption, creating a spectrum of biochemical outcomes. When hexosaminidase A activity falls below a critical threshold, GM2 gangliosides accumulate in neuronal lysosomes. These lipids interfere with intracellular trafficking and signaling, leading to cellular enlargement and impaired synaptic function. Evidence from histological studies shows characteristic vacuolation of neurons, while biochemical tests confirm elevated GM2 concentrations across affected tissues. Now, this mechanism forms the diagnostic basis for identifying disease severity in clinical and laboratory settings. Genetic sequencing reveals recurrent HEXA variants clustered within Ashkenazi populations, particularly the 1278 INSTATC and G269S mutations. In contrast, different founder mutations appear in French, Canadian, and Cajun groups, indicating independent lineage-specific events. Cultured neuronal models expressing these variants demonstrate sharply reduced catalytic efficiency, supporting the causal link between genotype and disrupted metabolism. Carrier frequencies reach their highest levels in Ashkenazi Jewish groups due to historical demographic isolation. By comparison, global populations show much lower prevalence, reflecting broader gene flow and larger effective population sizes. These distributions illustrate how drift, founder events, and social boundaries shaped the modern pattern of Tay Sachs mutations. As populations migrated across Europe and North America, characteristic HEXA variants expanded with them. Today, these patterns highlight how molecular defects interact with demographic history, offering a clear view of the interplay between biology and lineage over time. Founder effects emerge when new populations originate from a limited number of individuals, carrying distinct genetic variants. In the context of Tay Sachs disease, a small number of HEXA mutations became established within Ashkenazi Jewish ancestors during periods of reduced population size. However, drift amplified these variants by chance, not by selective advantage, producing elevated carrier frequencies over successive generations. Historical communities often maintained social and geographic boundaries that limited intermarriage with surrounding populations. This restriction reduced gene flow, allowing specific variants to remain concentrated rather than dispersed. Now, these dynamics explain why the same few HEXA mutations recur across Ashkenazi groups in multiple regions. Large-scale molecular screenings consistently show that 3 major HEXA mutations account for most carrier status among Ashkenazi individuals. By contrast, French, Canadian, and Cajun populations show different founder mutations, confirming independent demographic origins. The clustering of variants within each group reflects historical isolation rather than shared deep ancestry. Today, carrier frequencies in Ashkenazi populations remain several times higher than global averages. In contrast, populations with greater mixing such as most European and Asian groups, display low prevalence. These geographic patterns align with models demonstrating that drift effects diminish as effective. Population size increases. As communities migrated within Europe and later to North America, characteristic HEXA variants expanded with them. These patterns illustrate how demographic history, rather than selective advantage, shaped the modern map of Tay Sachs distribution, offering a measured view of how genetic variation persists through lineage and environment. Medieval Ashkenazi populations underwent several documented contractions that reduced effective population size. These events increased the influence of drift on rare alleles, including HEXA variants. However, the mutations did not originate during deep prehistory. Instead, their rise is consistent with later demographic constraints. Genetic simulations support the conclusion that bottlenecks between the 10th and 15th centuries produced conditions favoring the amplification of recessive variants. As Ashkenazi groups moved from the Rhineland into Eastern Europe, settlement patterns created small, semi-isolated communities. Now, each community served as a demographic subpopulation with its own founding individuals. This structure magnified local allele frequency changes, allowing specific HEXA mutations to persist and stabilize across generations. Historical documents describe repeated episodes of displacement, forced migration, and rebuilding. Population genetic studies show reduced heterozygosity and signatures of intense drift consistent with these records. By comparison, neighboring populations display more continuous gene flow and fewer drift effects, illustrating why Tay Sachs mutations did not rise similarly outside Ashkenazi lineages. Movement from Western Europe into Poland and Lithuania created new centers of settlement where demographic isolation continued. These regions show consistent carrier frequencies that align with known historical boundaries. In contrast, communities with later admixture show reduced prevalence, highlighting the impact of increased gene flow. Across centuries, demographic pressures shaped the landscape in which HEXA variants expanded. These patterns demonstrate how historical conditions can guide the persistence of specific genetic traits, linking molecular defects to broader narratives of migration and lineage. Carrier detection allows identification of individuals with a single pathogenic HEXA allele. Because carriers remain asymptomatic, the disorder persists silently within lineages unless systematic screening is employed. However, widespread testing shifts the demographic landscape by reducing the probability that two carriers will have an affected child. This approach exemplifies how genetic knowledge can modify disease prevalence without altering underlying mutation frequencies. Screening programs rely on two complementary methods. Enzymatic assays measure hexosaminidase A activity, which is partially reduced in carriers and absent in affected individuals. Molecular testing targets specific HEXA mutations, particularly the common founder variants in Ashkenazi populations. Now, comprehensive sequencing can identify additional rare alleles in diverse populations, improving diagnostic accuracy and expanding the scope of counseling services. Regions implementing coordinated screening, such as community-based programs in North America, show marked reductions in Tay Sachs incidence over several decades. These outcomes reflect the combined effects of early carrier identification, informed reproductive choices, and broader access to genetic testing. By comparison, areas without sustained programs report lower but stable carrier prevalence, demonstrating that screening influences incidence rather than mutation frequency. Increasing migration and admixture complicate ancestry-based risk estimates. As communities diversify, reliance on founder mutations alone becomes insufficient. Contemporary programs integrate both targeted and universal panels to address this shift, ensuring that screening remains effective across heterogeneous populations. Modern screening illustrates how clinical practice can reshape the expression of a genetic disorder while preserving the broader context of lineage and demographic history. It connects molecular insight with long-term patterns of population change in a measured and informative way.